Okay, so continuing with the cell, we're going to talk about the cytoplasm. Cytoplasm is a general term for material between the plasma membrane and the membrane surrounding the nucleus. It contains the cytosol, which is the intercellular fluid, as well as all the organelles. The cytosol, or intracellular fluid, contains dissolved nutrients, ions, proteins, and waste products. It differs from extracellular fluid in that cytosol contains a higher potassium and lower sodium concentrations than we find in extracellular fluid. It also contains higher concentrations of dissolved proteins than extracellular fluid. This gives the cytosol a thicker, more viscous consistency, and it usually contains small reserves of amino acids and lipids along with carbohydrates. This may also contain inclusions, which are insoluble materials. Insoluble means they can't dissolve in the water. The organelles, these are internal structures that perform specific functions within the cells. You have two types. You have membranous or membrane enclosed organelles. They are surrounded by a membrane, and they allow for storage as well as manufacture of substances. You also have non-membranous organelles, and these are in direct contact with the cytosol, the liquid portion. The first is the cytoskeleton. This provides an internal protein framework. It serves as the skeleton for the cell. It consists of thread-like filaments and hollow tubules or tubes that give strength and flexibility. And they're composed mainly of microfilaments, intermediate filaments, and microtubules. Microfilaments are the thinnest strands of the cytoskeleton. They're usually composed of actin, and they attach to the plasma membrane to the underlying cytoplasm. In muscle cells, they also participate in the actual contraction of the muscle. Intermediate filaments, they are intermediate in size, varying in protein composition, and they help to strengthen and stabilize the cells. And the microtubules, these are the largest and primary component of the cytoskeleton. They're found in all body cells. Hollow tubes that are built from globular proteins tubulin, and they give the cell strength and rigidity, as well as anchor organelles in place and form spindle apparatus during cell division. Microvilli, as we talked about, these are plasma membrane extensions containing microfilaments. The function of microvilli is to increase surface area, and you'll see this in areas where you have lots of absorption into the cell. The centrioles, the cytoplasm actually will contain two centrioles at right angles. Each centriole is composed of 9 microtubule triplets. The functions of the centrioles are involved in mitosis or cell division. They are essential for movement of chromosomes during cell division. Cilia, these are very long extensions of the plasma membrane that contain microtubules. There are two types, motile and primary. And the motile will use ATP to move substances across the cell versus the primary, which will act as a signal sensor. The function of these cilia is multiple motile cilia move materials over the cell surfaces. A solitary primary cilium can act as a sensor. Ribosomes, these are proteins and RNA that can either be bound to the rough endoplasmic reticulum, or they can be freely found scattered throughout the cytoplasm. And the main function of ribosomes is this is where protein synthesis occurs. This is the place where cells will make proteins. Proteasomes, these are hollow cylindrical organs of proteolytic enzymes. Proteolytic means an enzyme that's going to break down proteins. And the job of this is actually to break down and recycle that of damaged or abnormal proteins. The endoplasmic reticulum, this comes in two forms, rough ER. The rough ER has ribosome, so this is going to be involved in protein synthesis. The smooth ER this will synthesize lipids and carbohydrates. And again, the overall function of the endoplasmic reticulum is the synthesis of secretory products, meaning products that would be secreted from the cell, and intracellular storage and transport. So though, again, there's four main functions to the endoplasmic reticulum. That is synthesis of proteins, carbohydrates, and lipids, storage of materials, isolating them from the cytosol, transport of materials through the cell, as well as detoxification of drugs and toxins. The smoothie R is called the smoothie R because it has no ribosomes. It synthesizes lipids and carbohydrates, such as phospholipids and cholesterol from membranes, steroid hormones, glycerides, and glycogen in skeletal muscle and liver cells. The rough ER has fixed ribosomes on the membrane. It is the location of protein synthesis. Some proteins are packaged into membrane-bound sacs, and these sacs, or transport vesicles, will pinch off from the tips of the ER and deliver the proteins to the Golgi apparatus. The Golgi apparatus, this is made up of membrane disc called cisternae, and there are three major functions to the Golgi: modify and package secretions for release, renew or modify plasma membrane, package enzymes within the lysosomes. Lysosomes, these are vesicles that are found throughout the cytoplasm, and they contain digestive enzymes. The functions of the lysosomes are intracellular removal of damaged organelles or pathogens. Peroxisomes, these are also vesicles very similar to that of lysosomes, and their main function is the catabolism. Catabolism is the breakdown of fatty acids and other organic compounds, such as lipids, maybe carbohydrates, starches. The peroxisomes will also neutralize that of toxic compounds that are also generated through any of these processes. The mitochondria, this provides the energy for the cell in the form of ATP. They have a double membrane. The outer membrane surrounds the organelle. The inner membrane is formed to form cristae, which increases the surface area exposed to the fluid matrix inside the cell. Inside the mitochondria. Enzymes in this matrix will catalyze energy-producing reactions. The first step of breaking down glucose, which is in a process called glycolysis, occurs in the cytosol. The remaining steps of aerobic metabolism, or more commonly referred to as cellular respiration, these processes will occur in the mitochondria. Aerobic metabolism produces about 95% of the cell's ATP, or energy. And for humans, this is a very essential process. If your cells are not able to undergo mitochondrial activity, those cells will die. The nucleus, this is usually the largest structure in a cell. It's referred to as the control center of the cell. This will dictate cell function and structure by controlling protein synthesis. The nuclear envelope is the membrane structure that surrounds the nucleus, and it separates the cytosol, which is what's found on the outside of the nuclear envelope, from the nucleoplasm, which is essentially the cytoplasm inside the nucleus. You have these little pores called nuclear pores, which allow movement of substances into and out of the nucleus. So here's our nucleus, and again, inside we're going to find all of the DNA. For humans, that would be 23 pairs of chromosomes. There's also another structure within the nucleus called the nucleoli, or nucleolus. And the nucleolus is where you're going to synthesize ribosomal RNA, or ribosomes. And if you remember back a few slides, ribosomes are essentially important for the production of proteins. Within the nucleus, we'll also find our chromosomes. As I mentioned, the human body has 23 pairs of chromosomes. Okay. So information storage in the nucleus is your genetic code. the chemical language of the cell. It contains information for protein synthesis in the form of a nitrogen-based sequence, which is what forms DNA. It is arranged in a sequence of 3 bases, or 3 letters, so-called a triplet code. Each triplet code represents a single amino acid, and amino acids are what make up proteins. The gene is a functional unit of heredity. Each protein coding gene contains all the triplets needed to produce a specific protein. Genes are normally tightly coiled with histones, which are proteins, keeping them inactive. In order to make proteins, you have to go through 2 processes called transcription and translation. Transcription is the production of a single strand of messenger RNA from a single strand of DNA. This process required to get DNA blueprint for protein synthesis from the nucleus out to the cytosol, where the ribosomes are. So what happens is when you make the messenger RNA, we're essentially making a copy of the gene, because the DNA cannot leave the nucleus. Remember, protein synthesis occurs on the ribosomes, which is going to be found on the rough ER or in the cytoplasm. So you're going to make a copy of the gene, and that copy of the gene will be a messenger RNA, which will leave the nucleus and go to a ribosome. When the messenger RNA leaves the nucleus and goes to a ribosome, it will undergo translation. Translation is where protein synthesis using the new messenger RNA codons to signal the assembly of the specific amino acids. And this will generate the protein structure. Finally, cell divisions. One of the important things of a multicellular organism is that new cells have to form. This occurs through a process called mitosis. Mitosis is nuclear division of your body cells. And these will derive from stem cells, and it essentially will make 2 daughter cells. Apoptosis is a genetically controlled death of cells, so sometimes cells need to die in order for the goodness of the organism. Meiosis is a different process. Meiosis is a process in which you're producing your sex cells, the sperm and the ova. The difference between a cell made by mitosis and a cell made by meiosis is the number of chromosomes. A human cell has 23 pairs of chromosomes. When it divides by mitosis, each daughter cell will end up with 23 pairs of chromosomes. So they'll make copies, and you'll have 23 pairs in each cell. With meiosis, you actually go through another round of division where each sperm and each egg will only end up with 23 chromosomes, not 23 pairs of chromosomes. The reason for that is when the sperm meets up with the egg, you have 23 pairs meeting up with you have 23 chromosomes meeting up with 23 chromosomes, forming the zygote, which is what happens when the sperm and egg fuse together. And that new cell that forms will now have 23 pairs, which is what defines a human. Cellular specialization due to gene activation or repression, meaning cells will differ based on what genes they have turned on or off. And differentiation occurs when specific genes are turned off, leaving cell with limited capabilities. And a collection cells with specific functions are called tissues, which will be our next topic of discussion.